all the things that you need to know about the COVID, about pandemic, and what is Pathogen. Anyway, hi and welcome to my channel. My name is Dr. Roy Yosevich, and in this channel, I host and speak with the most interesting and influential people and scholars from all around the world to discuss science, philosophy, theology, artificial intelligence, and creativity. If this is your first time on this channel, please consider subscribing and hit the bell button. And my guest today is Professor John Tregoning from I just wrote it, from the Imperial College at London. John Tregoning is an associate professor in respiratory infectious in the Department of Infectious Diseases at Imperial College in London. And most recently, he published this great book, Infectious, Pathogen, and How They Infect Us. So I can't read it because it's upside down. Pathogen and How We Fight Them. So Professor Tregoning, thank you so much for coming. How are you today?
Very good, thank you. And thank you for inviting me to talk to you.
Okay, so like I said before, I have a problem with you since your research about diseases and pandemics and all the other things is extremely important, is very interesting, and of course it's relevant. But your meta-research about how you published your book, and how do you become better in academia might be even more relevant to my audience. So with your permission, we will start with the first and go to the second, okay? So first, this is a great book, and one of the things that you wrote about your book that you mentioned the word serendipity, because you are a professor of infectious diseases, and I think that being in the COVID area, being a professor in infectious diseases is like, sorry for saying it, it's like a dream come true, yes?
That's an interesting question. It certainly, yeah, it's changed the way, well, no, it's really hard to explain.
I'm sorry.
No. It sort of changed, some of it has changed the way we've thought about. So some of it is like, this is what we expected to happen. This is how we thought things would happen. And then some of it is like, well, that's not what I thought would happen. That has changed. So it's it is interesting, but there's that sort of curse about, may you live in interesting times, where actually when you are in the thick of it, and it is quite exhausting, but yeah, fascinating, but exhausting.
So I just want to mention one thing, that may you live in interesting time, consider a curse in Chinese literature or in Chinese tradition. So interesting is only nice in retrospective, not during what you did. So let's start and dive into the science. I want to read out loud a quote from your book, and I think that this will be the best place to start. A disease is not the same thing as the thing that causes it. Diseases refers to symptoms. So COVID-19, which we know as the COVID virus, is the uncatchy name for disease caused by similarity uncatchy SARS-CoV-2 virus. So what do we need to know about the distinction between diseases and the things that causes diseases? This is just like phenotype and genotype, or something else?
It's something more. I think if we think about the pathogen, which is the agent that caught the infectious agent, so it could be a virus, it could be a bacteria. If we're thinking about SARS-CoV-2, it's a virus. That agent, you can be exposed to that agent and not get sick, or you can have the agent, you can have the virus, it can replicate maybe in your nose and make you mildly unwell. It's only when it stacks so that the virus may be more virulent, your immune system may be compromised in a way, or your lungs are compromised in a way, that you get these sort of severe disease. And that's why I think, going back to your saying, you know, living through this interesting time, is you see this whole spectrum of disease in lots of different people. And because we're measuring who's getting infected, we're seeing infection, so people with detectable virus everywhere, but we're not seeing sick people everywhere. And that's changing with the vaccines as well. So people are still maybe getting infected, but they're not getting as severe sick. So you've got these kind of two things separating between agents that can make you unwell if you're that way sort of predisposed versus the kind of the disease that can come. And just one final bit on that, the disease actually can be quite similar between different pathogens. So if you get a bad flu infections into your lung or a bad SARS-CoV-2 infection into your lung, you'll have a similar kind of symptoms. You'll present with difficulty to breathe, pneumonia. So there's similarities in the kind of body's response, especially when things are going wrong, regardless of the virus.
Okay, so let's say that I'm having a PCR test, and according to the PCR test, I'm COVID positive. So, but I have no symptom whatsoever. Okay, and this is what we call in Israel or in the world non-symptom. We call it asymptomatic patients. So they can infect other people, but they don't get the symptoms. They don't get the cold, the flu, the difficulty in breathing. But what science know about what distinguishes people between getting the symptoms and not getting the symptom? Is it pure luck or something inherent different in their immune system, in their biology that make them immune to the symptoms of the disease?
So there'll be, I guess it's like maybe like a computer, it's like a gating thing. There are multiple gates through which things have to go in order for the disease to happen. So the first is probably the amount of virus you breathe in. So if you breathe in one tiny virus particle, you're less likely to get sick than if you breathe in 100,000. So that's the dose is then important. The second thing might be like the time of day. So we know that the immune system is more active at different times. The circadian rhythm, it changes. So it may be the time of day that you're exposed, or how tired you are, or how dry the mucus in your nose is. So there's lots of kind of barriers, and the virus has to kind of overcome all of those barriers. So we know things that will tend to predispose people. So if you have COPD, so the condition that follows smoking throughout your life. So if you've damaged your lungs through smoking, you're more likely to get sick. Or if you have diabetes, you're more likely to get sick, or a heart condition. So we know things that may predispose people to get more sick, and age seems to be the major driving factor. So those things, all of those things kind of add up. And I don't think what we don't have is enough knowledge to be able to say, if we took me and just measured all of those bits and pieces and put them into an algorithm, we could say, this is how sick you'll get. And that's the kind of, that's probably quite a long way in the future, but that would be a fascinating thing to be able to do, just to kind of put all these variables in and say, this is how sick you'll get.
But this is basically a chaos theory. It's a complex dynamic system that have many, many parameters. And you said it for yourself. You said it even the time of the day. So let me get it straight, because it is fascinating. And this is the first time that I hear this. So I might get the virus at, 10 a.m., when I'm fully alert and I am not tired and I ate properly. And then my body going to not let the virus go into the gate. And then even though I will get the virus later at 10 p.m., yes, when I'm very tired, the fact that the initial encounter with the virus when I was very healthy means that I developed like a natural vaccine.
I think it's probably more just it's repeated attacks against the same thing. And if the attack in the morning bounces off, it doesn't necessarily mean that the attack in the evening will also fail. So I think I think what it tells us in terms of exposure is the more exposures you have, the more likely you are to get infected. So that's where masks and the non-pharmaceutical interventions and good ventilation help, because you're just reducing the number of chances that the virus gets to roll the dice and get into the body.
Okay.
But yeah, I think you're right. I think there is this sort of chaos element to it. There's so many variables that it is very hard to predict But I don't think, but then coming back to the public health side, everything we can do to put more barriers in the way is going to reduce the chance of disease. So vaccines and good hygiene and good underlying health, all of those will help you in kind of swing the balance in your favor.
Okay, so let's move on to chapter 4 in your book, and it's microbiome. Now, I had on the show Professor Rosman from Weizmann Institute in Israel, and he is a cancer researcher, and what they discovered that even that the microbiome, that the germs in your, I don't know, in your stomach has a great impact on what we called cancer treatment. So please, shortly, could you explain what microbiome is and how it affects your possibility or chances to get diseases in the context of the COVID?
So the microbiome refers to all of the mostly bacteria. Let's say bacteria. So microbiome refers to all of the bacteria that live on or inside your body. So you have bacteria on your skin, you have bacteria in your nose, but the majority of your bacteria live in your guts. So and they help you digest food. They help you stop getting secondary infections. They help particularly metabolism, but they also seem to train your immune system. So if you count the number of cells of bacteria in your body versus the number of cells, there's about the same number. So we're in some views, we're like a massive taxi for bacteria rather than just a kind of human body. Wow. What do they do for COVID? There does seem to be some correlation between maybe certain bacteria types in the gut and better immune health. And it's certainly, if you have probably what's best known is it's a bit like a jungle. The lower the diversity, the less healthy the ecosystem. So the more different types of things.
Again, the lower the diversity of the bacteria, the different kinds of bacteria in your gut, in your body. So the lower the diversity,
the more probably the worse outcomes are. You want to have like a broad range of different things that can respond to different outcomes. But it's still very early in our understanding of how these work. And they're difficult studies to do and to do well, because it's quite easy, because there's bacteria everywhere. You know, there's bacteria on my coffee cup and it's quite, and in my science for pets. And so it's quite easy to get contamination and kind of misunderstand things. So I haven't seen anything like a smoking gun, a thing that says this is the single bug you need to be better. But it's more diversity, eating more healthily will all add to that and kind of lead to better outcomes.
From what I understand, and I spoke with Ravid, it seems that the microbiome is like an explanation mechanism, but not a prediction mechanism, since there are so many factors. Even what happens to you during birth and in the birth canal, and the fact that you have a dog or you don't have a dog, and your environment. So the microbiome is affected by so many factors that you cannot control. So it's like a unique fingerprint of the person, and your ability to change it or to alter it in a specific manner, just like you said, is very limited nowadays. Am I correct?
Yeah, and I think it can change, and it's quite fluid. You know, I think if you measure it at different times of the day, it changes. There was an amazing paper which linked, coffee intake and the type of bread you eat, and there are so many factors in changing all of these things. I think to make sustained changes in it, I think is very targeted changes. It is very difficult. I think where it has been really interesting is in this fecal microbiome transplant. So what we do know is that if you take a lot of antibiotics, you kill a lot of the microbiome in your guts, and that can have long-term effects and make you more susceptible to a bacterial infection. So if you can reseed it after the antibiotics, you then protect yourself against these secondary infections. So keeping it kind of relatively diverse seems to be healthy.
By the way, and I think that many people say there is a joke in Israel that flu, you can cure it with antibiotics in 7 days, but if you don't take antibiotics, it will go after 1 week. So basically what we say is that the natural immune system of the body is extremely efficient. And many people say that even in the context of the COVID, where people get the vaccines, the natural vaccine after being ill, after being sick, and they develop the vaccine, this vaccine is much more sustainable for different kind of mutations. So is it true that the natural vaccine or the natural immune system will be better in certain aspects over the Pfizer vaccine, for example?
So I think the first important thing is, if we go back to our not understanding who's going to get sick, is that if you're saying the infection with the virus, you are rolling a massive dice of because you might get sick. So that infection could very easily, you may have a genetic susceptibility that you didn't know about, and it could put you in hospital. So that's a very high risk strategy. The second thing is the viruses make anti-immune factors. So the way that viruses infect you is by turning off your immune system. And we think that some viruses, like coronaviruses, actually dampen the immune memory. So they make a protein that targets the immune memory and basically shuts it down, makes your memory less good. So on those levels, natural infection is probably less effective because you've got the high risk and you've got the kind of a rapidly diminishing response. What the vaccines do is that they give you the immune memory without ever being exposed to the pathogen. So you never get sick, and you might get the mild infection, but you're never going to get the severe disease. And the vaccines are protecting against severe disease. And I think they give you a stronger, more controlled response against infection.
But in Israel, we see that we had like the first shot of the vaccine, and then the second, and then the third, which we called the booster. And then we have a Omicron, which is like the fifth or the fourth. I don't count, yes, the fourth version of the virus. So it like we just tweet the very specific modification of the virus each time, and each time the immune system, it like diffs and cops, yes? Each time the virus become more and more sophisticated. So what about this? Isn't like are we going to be another vaccine and another vaccine for the next 10 years? Or what do you think is going to happen?
Yeah, so it is an interesting question. So with the vaccines and the booster, so the 3 doses that one would have had in Israel, they're all the same thing. They've not changed the vaccine at all between those 3. So it was targeting the original strain that emerged in 2019. And it's basically what happens is each time you get vaccinated, it pushes the amount of antibody up a level. So the first, you're here, you have your second one, it goes to here, you have your third one, it goes up again. And that comes back to that idea I was talking about putting more gates in the way. The more antibody you have, it will soak up more of the virus. Even if the antibody is less efficient because the virus is changing, you are still soaking up most of it, and that will, again, reduce your risk of severe disease. The virus is evolving, and that is natural, virus, because it's what's called an RNA virus. So it's the genetic material in it is a bit more leaky. So when it's like if you photocopy a photocopy and you keep on copying, instead of going back to the original, you make more and more copies, you get more and more errors in the document. So the viruses make more and more errors. The more people who are infected, there's more chances of these errors happening. And some of those errors, most of those errors will kill the virus. It'll be bad for the virus. A few of them are going to give it a selective advantage. And what we don't know is what's the selective pressure, because we don't know whether if it did emerge in Southern Africa where the vaccine coverage is low, it's probably emerged because people had previous exposure, natural immunity. So if vaccine coverage was universally high, there would be fewer viruses and fewer chances to mutate.
Yes, but those viruses can overcome the vaccine now, because if all the people in Europe, in Israel, or most of the people in Europe or Israel or in the US have the vaccine, so any modification that will survive this stage means basically that it can survive the gates of the current vaccine. Am I correct?
No, because it's not binary. It's a continuum. Like the best graph I've seen about protection is like basically a sort of S shape, but with very noisy intervals. So some people are high, some people are low, and the vaccine is going to give you noisy protection. It's not going to be binary protection. So I think it will still give you protection, and the more antibody you have, even though they're suboptimal, like having 10,000 suboptimal antibodies is better than having 1,000 suboptimal antibodies. So that it will continue to give protection. What may be required is new, like a bit like with flu, where the virus does change season on season. We may get to a point where we do need seasonal targeted boosters against this virus, but we just honestly don't know at this point, because there's not been I mean, there are endemic coronaviruses. There's like human coronaviruses out there that do circulate all the time and cause kind of colds and things. So I don't think we know what is required next. I think it's prudent to look into making or testing new vaccines against the new strains as an insurance policy as vaccine immunity wanes.
Okay, so with your permission, let me go to another quote from your book, which is, while infectious have coexisted with humans throughout our evolution, pandemics are a feature of civilization, okay? And the word pandemic means all people, yes? People need to be living close enough proximity to allow the infection to spread within communities and have good enough contact between communities to spread it more broadly. And we have seen it, I think, vividly during the COVID pandemic, where we started in China, and then after 2 or 3 months, it spread throughout Europe, Israel, and the US. So are we, in a way, if we take the Spanish flu from like 100 years ago, our world today is much more connected. And therefore, the Spanish flu influence only in certain borders. So basically, what you mean is that we are much more susceptible to pandemics and disaster like this as we get more and more connected, as our world become flat, as Thomas Friedman put it. Yes?
I think the risk of the spread is higher, but the tools that we have to deal with them are better. I think that both things are developing at the same time. The sort of, this Matt Ridley's idea about the spreading of ideas and how quickly ideas spread. What if you compare back to 1918, we didn't know that influenza was caused by a virus. We didn't have a vaccine. We didn't have the supportive like ICU. We didn't have what's the word I'm looking for? Like the steroid drugs that are very useful in the kind of severe diseases. So the knowledge has increased at the same time as the kind of human population kind of increase in number. But so yes, in general, we are at greater risk because of the interconnectivity.
And we didn't have a test state, which was Israel, that it's like a giant lab experiment, never been conducted before. It was like a test state that Pfizer said, okay, let's test the vaccine on the people of Israel. So I think that it gave us a lot of information, no?
I mean, the vaccine was tested in all of the vaccines had a massive phase 3 efficacy trial before they went into countries. So they were tested globally for safety and protection before they were then licensed into different countries. So I hope by being an early adopter, Israel then generated more of the early data, but the test happened. I don't like the phrase test state. I don't think that's a fair description. I think.
Okay. I'm sorry.
No, it's fine. Sometimes these things sound odd.
I'm from Israel, so I just want to raise to my country. This is basically it. Okay, but there is a chapter from your book And this chapter says, which leads to the big questions. And he says, why don't we get sick all the time? And this is a very important question, because if you get the bacteria, if you get the disease, if you get the viruses from the air, and each breath of air, it's approximately half liter of air, how come that we don't get sick all the time? This is a very important question.
Yeah, no, I think it's amazing. We've done some research where we were looking at the number of, we swabbed 200 children and measured viral infections in them, and 20% of them had what would be called a viral pathogen, but they weren't sick. So there are these viruses and bacteria out there all the time, and I think it speaks to how good our defenses are in protecting us, but also how well evolved, and that's a bit of a clunky phrase, but how the viruses have evolved to infectious, the kind of things they do to get past these barriers. And when you sort of look at some of the microbiology research where people examine single proteins in bacteria and show that the single protein has all these different functions just to help the bacteria win in its battle, it's a remarkable kind of evolutionary arms race. I think I talk about the image of the Red Queen hypothesis in Alice in Wonderland, where they're both running at the same speed, but nothing changes. And I think it's some of that. It is, yeah. And we don't have the answer. And I think that is what drives my lab's research, is to think about, well, what is it that happens in those times when you do get infected?
Because it's when you think about it, it's like it's so unprobabilistic. The probability of not getting sick is so low that how come? It's like a living miracle. So I want to go, you, from my perspective, represent the academia, the university. And many people say, listen, we invest tremendous amount of money in academia, NSF, ISF, Horizon, giants of billions of dollars in euros. And when we needed a straight answer from academia, and all the academia joined together to give us something, the answer wasn't there. And we got the answer from Pfizer, which not part of the academia. It's not it. So my question, and I spoke on the channel with several biologists that mentioned this very thing, that how come we didn't get the answer, so do we need to rethink our biology and microbiology department after the pandemic, that we didn't get as the public, as government, what we needed them after so many years of investing tons of money?
Yeah. So I'll come back with a straight no on that. I think that's a misinterpretation of what happened. I'll give you a number.
Okay, why big no?
So the big no. So let me go through a number of examples why I think the academic contribution to the COVID pandemic is enormous. The first is, if we look at the Moderna vaccine uses what was called a pre-fusion stabilized form of spike. So the spike protein, that's the bit of the virus that the virus uses to get into our cells, and the thing that the vaccine targets is basically like a spring-up umbrella. It has a compact form that it lives in on the virus, and then the open form that it uses to get into cells. That compact pre-fusion, the form before it opens up, is the more immunogenic, is the one that you want to get protection against. Understanding that knowledge came from a group at NIH by Barney Graham and Jason McClellan, studying as an academic group a different virus. And their knowledge, their 20 years of research and knowledge, went into the design of the Moderna and the Pfizer vaccine. So without that academic group, we wouldn't have the Moderna vaccine. The second part of the Pfizer vaccine was that it was developed by, it's made of mRNA, and mRNA, when you look at, when your cells see it, says this is foreign, and normally your body would say, because this is foreign, like because it thinks it's an infection, would stop the mRNA being turned into proteins. So how I need to drill down. It's basically mRNA encodes the proteins. Your body would turn that off. There was a group called Kathy Carrico, who works at BioNTech now, spent 20 years as an academic researching how to trick the body into making mRNA vaccines. So the two key bits of underpinning knowledge into the mRNA vaccines came out of academic research. And both of them were kind of quite somewhat niche and wouldn't have been funded in industry. I mean, the third one, if you look at the AstraZeneca vaccine, that is all built out of research done at Oxford University over a 20-year time span. So all of this knowledge, what happened in the last year was basically the tip of the spear of 20 years of research. And the 20 years of research came from academia. So without that open blue sky science space, we wouldn't have the knowledge to build the things that we need in the last 2 years. So I think no, academia can't make a factory where you make vaccines, but yes, they can make the knowledge that then feeds into the factory at the end.
Okay, so with your permission, because I think, and from the passions that I get from you, this is an important question. And let me tackle this from another perspective, with your permission. And the other perspective is no vaccine and no scientific progress is possible without research, without science. No question whatsoever. Now, the only question is, what is the best or the most efficient platform for specific scientific progress? In the field of AI, nowadays, it is the big tech. All the big research is conducted in the big tech, or most of the big research is conducted in the big tech. In the area of the transistor, it was Bell Laboratories, okay? The BJT, the transistor, the diode, all the other things were conducted not in the university. It was conducted by great experts, great scholars, great professors, but not in the platform of the university, okay?
Wasn't Bell Labs run like an academic department? It didn't have a specific...
Bell Lab ran like academic department, and Google Research ran like academic department, and Facebook Research ran like academic department very well. But there is something different, because Google fund itself and Facebook fund itself. So it is extremely important to get the distinction. Now, we say, okay, we need basic science, and basic science is unprofitable, okay? Because you just aim to a very long, very far away target, and then you try and maybe you get it and maybe you don't. So maybe we want academia to focus mainly on basic science, just like you said, like 20 years research that eventually leads to what we need. And of course, without Maxwell equation of electrodynamics, we wouldn't have anything. It is important. We need the academia, but the platform that we call academia, universities, grants, etc., is not efficient in delivering things to the door, to delivering the next vaccine, to delivering something that is working. What about this perspective?
No, I agree. I think academia has. I think it's an ecosystem. You can't have industry without academia. The two things, and so I think that there's kind of there is a mergy place in the middle of early phase 1 trials. But to my mind, academia is about generating a number of ideas, many of which won't work. Therefore it's the kind of, it's almost like the mutation that drives evolution. So you're having all these mutant ideas come up, most of them fail, but some of them will then feed into the industrial pipeline. The other thing I think academia does, which is really important, is that it trains the people to go into industry. It trains the people to go into policy. So we can use research as an educational tool. So at that point, you're then getting new ideas that may or not be important, but also you've trained somebody who can then go and be a scientist thinker in any number of different jobs. And I'm strongly, I think what I do as a supervisor is training scientists, not training academics. And I look at the kind of people who've been through my labs. One's a medical doctor, one works for the government, one works for a funder, one works for a science writer. So there's lots of different paths that I think the academic model then contributes as well. Ideas and people, I think, is what we generate.
But I'm professor of computer science, and as a computer science, many companies in the industries says that learning to program in academia is not good because academics don't know how to really program something that works. So in other words, we train our students to become better scientists or better thinkers, but we don't train them, we don't give them the proper tools, and those proper tools are given to them in the companies and the corporates that they are working. And I would argue that this is similar in Pfizer, that if you that what you get when working in such a company like Teva or Pfizer is different, and in some aspect inherently different than what we get in the academia lab. So with your permission, let me ask you, do you think that something in the model of training, in the model of syllabus, in the model of funding academia in your department should be changed after what we know after 2 years of the pandemics? Or would you say, no, just keep it the way it was?
So in terms of training, and I think universities are moving there, is we are accepting that the knowledge of the world is in our mobile phones now. You don't have to learn the Krebs cycle off or whatever it is. You don't have to learn that into your head anymore because you can look it up. But you do need to be learning critical thinking and experimental design and planning. And those are the skills that I think in terms of the education is what we are doing both at a undergraduate and a postgraduate level. In terms of funding, I don't know. I mean, I of course would argue for more funding.
I won't tell, John.
yeah.
It's just between the two of us.
I think it's been fairly. I don't think academia failed in the pandemic, because I think across the board you've got the epidemiologists and the modelers and the vaccine people. So I think it by and large works. And I don't know. I think it's a good return on investment.
Yes, because in the last chapter of your book, you say, and you speak about, the balance between companies and academia and what they should do. But with your permission, because it was extremely fascinating. But I want to move on to the second thing. And what we talked about so far was, how academia influence something that is relevant to the public. And you most recently published this book, the books that we are talking about, Infectious, Pathogens, and now we find them. And something very interesting happened. You also wrote a column in Nature Careers, how I wrote a pop science book. Now, and this is extremely well written, very funny, very funny. And first of all, what is Nature Career? It's like a Nature article that you have there, but it.
So Nature, the journal has kind of two bits. The main journal has two bits. It has the kind of magazine-y, newspapery side of it, which does the articles and the careers advice, as well as the scientific writing. And I've been working with the team on the career side for a number of years about writing different aspects of what it's like to be a scientist, and how and different challenges in academia and how to overcome them. So I was very lucky during the pandemic because I wrote through them. I wrote a weekly diary of what it was like to live as an academic through COVID, and that was a lot of fun. But it was quite a challenge because I was writing, like, every week you'd be writing one article, editing the one from the week before, and thinking about the next one. So it was a very different sort of intellectual challenge to the normal job.
No, but when you published in Nature Career, it's not like published in Nature. So or yes?
Well, apparently it appears, it's cited on PubMed as a Nature paper, but it's not. No, it's not a primary research paper. But
it might be my only chance to go into Nature. So exactly. This is why I'm asking. Okay, but we know that Faraday wrote the story of candle as addressed to youth. We know that the publisher of The Origin of Species told Darwin, make the book clearer. I want to address the public. But we know nowadays that scientific articles are written in a way that are inaccessible to anyone outside the realm. And this is causing, it's like it's a bad policy, because we separate science from society, we separate science from the people. Most people who are not in the field or not in academia have never accessed Google Scholar before. I know it's like mind-blowing, because we work with Scholar all the time, but just when I became a graduate student, I knew that such a website exist. So, and I think that this is part of us to blame, the scientists, because we don't write clear enough. And I think that, and it is very, what I didn't like about your title is this is a pop science book. The pop, because it is fun, it is full of anecdotes, it's funny. And why being fun, full of anecdotes and engaging means pop science book?
So I think the scientific paper is a very specific tool. It's a peer-to-peer tool, isn't it? It's not about engaging public. What I've always tried to do then is write a separate article as a lay companion piece that then explains the science in a way that, you know, my parents and my family could then understand and see. So I don't think it's possible to change the scientific paper in a way to make it more, because you're trying to communicate information to a different audience. So I think what we have to do is get better at communicating to both audiences, and also then to value the communication to a broader audience. I think if you take the drivers of the academic career are bring in money, produce papers. They're not about how well you communicate your science. So it's something that we do on top of our day jobs rather than kind of as part of our day jobs.
Yes, and this is bad because it's like grants are not part of your papers. Now, I wrote four lay audience of 4 books, which I invested my time and my life in those books. And those books, those books, or part of them, influence the lives of people much more than the papers that I've published in Google Scholars. And let me disagree with your permission, because you said you need to address 2 kinds of audience. You need to address the peer review, your peer scientists, that you need to tell them, listen, this is what I done. This is what the results are. Please continue from there. And this is how science build, one brick at a time. Okay, which is great. But I will argue that this is not the case. And we see in my discipline and in electrical engineering that nowadays people try to be more complex, more vague. Instead of taking very simple equation, they just fill some of them fills the paper or fills the page with unnecessarily mathematics, just to make sure that if the paper is hard to understood, it must be very deep. And I will tell you even more that the tendency today, and you also know it, is to publish short and shorter papers because people don't have the time to read so much. And so, in other words, what you describe is like the utopia. Okay, I need to tell you, okay, what I've published and why it is important, and so you can move on. Yes, it's like a stick race. I don't know how you say it, like a pole race. But this is not the case. And many articles, many scientific articles are extremely hard to read. So what can we do to change it? You change it in your blog, and I change it in my writings, and I try to be, better in my writing. Let me tell you a story. Randy Pausch, the distinguished professor from Cornell University, from Carnegie Mellon, who passed away from cancer. He did it his tenure. He did it sabbatical in Disney Imagineering. And after Disney Imagineering, after 1 year of Disney Imagineering, they had to publish the paper. And the guy from Disney Imagineering said, listen, this paper is so pale, add the picture. And they added the picture before the abstract. They added the picture before. And the people at the journal, well, are they allowed to do it? Why do we need to even say, are we allowed to add picture? Why can't we ease the access of our papers even to our peers? Because it's hard even to our peers to get to read our papers. What do you think?
No, I agree. I think there's a lot of bad science writing. But it's hard, right? It's taken me 20 years to be able to write a science paper okay-ish. It's a skill. And you have to practice and you have to get good feedback. And I think you're right. I think there's such a volume out there that it doesn't give the editors the chance to edit. I think if you go to some places, I had a very positive experience with a reviews article where the editor edited every line and worked with me. So I think when editors are acting in like a newspaper editor, you get a much better product than when you get an editor just acting as a gatekeeper in terms of the science. So I think
you get much better training.
Yeah, because you can see what works, and if you get some feedback and say, don't just say the same word over and over again. I think there is and there's also this. I don't know if you see it, but I feel sometimes people, like you say, obfuscate. They use complicated words where a simple word would do, and I think, like I just did. So I think there's a bit in writing where it's just say it was rather than and so we saw that this thing. I was like, well, that's just not good writing.
Yeah, it's not used. It's like academia. It's like I don't say I used. I say I utilized, and we don't say it was a passive form. And if you see, Stephen King has some rules about good writing, and Stephen King is a very good writer. Yes, he's like the most read author in the 20th century. And basically, I think all the things that he described as good writing are like in academia, are like in journal writing. And you see all this complex sentences. Just a second, what we are trying to do. Okay, I want to do this. And I add some humor, add some quotes, add some, all the quotes and the anecdotes that you add to your book are so important because they let me hold. It's like a hook to get the idea, to get the theoretical part. And it's extremely important. And we rarely see them in scientific writing. We see them only in the best scientific writing, but not in all of the scientific writing. So my question is, because you wrote a book about science, you said in one of the tips that save many factual data, many anecdotes as you can, because they will spice up your writing and will be important as well. So my question to you is, how do you save and file your anecdotes? Because we get like many anecdotes and we read something that, wow, listen, this is good and this is good and this is good. But how do we save them in a way that we can restore them efficiently afterwards?
I didn't have any system. They were just things. I think they were just things that made. I had enough of. They're either things that had made me laugh previously, and I really remembered them, or I had enough of a stub that I could then search them on Google. So I think my search terms are probably quite weird at the moment. Like, what's the most? What's the longest tapeworm, or what's the most? How much? Because I remember going to talk about somebody doing human deliberate cholera challenge, and he was talking about the volumes of fluid that came in. I couldn't remember the exact number, but I could then piece it together. It was a bit of a kind of detective work to just try and find the bits from my brain from earlier. So maybe writing them down would have been better, but I just kind of relied on my imperfect memory.
Now, another thing that you said that you had to write 90,000 words, and you said, okay, 90,000 words, meaning basically 3,000 words each week. But when you have a diary, yes, like you kept track of the words that you were writing each week, and most of the weeks, the total numbers of words were less than 3,000, and which became that the weekend became very, very intense. Now, in retrospective, what could you have done differently? And what tip could you give to reach this like 3,000 words? Or do we need to think it otherwise, think about it differently?
So my friend and the sort of writing mentor who got me into it is Professor Dan Davis at Manchester. And he's written 3 books. And I know that, certainly he does take sabbaticals and just steps out and goes and does that. And lots of my friends were like, well, I don't know how you're juggling it. Maybe you should just go and do it. And so maybe it would have been better to have taken blocks of time, of being a bit braver about saying to my line manager, can I take these blocks of time as sabbatical and take it off? But there was something about the pace and the having to do it that meant I didn't ever have time to get bogged down and worried about it. So because it was so like you have to do it, there was no writer's block because there was no time for writer's block. So I don't know. It would be interesting to try it the other way, where I have like a luxurious, month in my calendar where I can just write, but I'd be worried I'd just fritter it away.
No, but deadlines are extremely important. I think that Terry Pratchett has a saying, I love deadlines and the sound that they make when they pass by me. Something like this. Now, another thing that I'm curious about, because I also write. Now, this is your field of research, your field of expertise, but sometimes during writing, during the work of a book, you get to a niche that you are not expert at. And now the question is, how much do I need to dive in and do research in order to elaborate my writing, my understanding? Should I add, or in other words, should I add another page or another chapter or another section? I started my book about artificial intelligence, and I said, okay, I need to have a small chapter about human intelligence, because artificial intelligence basically want to simulate human intelligence. And I start reading about human intelligence just to have like 20 pages. And eventually it came out with 400 pages because the subject was so interesting, so immense. I couldn't summarize it in 20 pages. So I had 400 pages. So I'm sure that this thing also happened to you. So what's your take about how much dive into a side niche?
Yeah, so I just slight different answer to start with. The thing I found the most challenging were the bits that I knew most about, because I couldn't work out where to crop them. So the immunology chapter does run on a bit when I was much more strict on the ones where I knew less. The second thing that helped was that I have access to experts in all of these fields because I work with them. So I could just send a chapter to somebody and say, have I missed anything on there? And the third, I don't know. I think I went as far as I was interested, and then I could just pick out the funny and the interesting and the important. So I'm sure there are things I've missed, and I'm sure for people, like I had friends who were like, oh, you didn't mention this pathogen. I was like, no. Have you seen Blackadder, where he's writing the dictionary? There's a British comedy from the 90s where he's writing a dictionary. And at the end, they say, he's like, oh, do you want a sausage? And he's like, sausage? Because he's really angry because he's missed the word sausage. And there was a bit of that when somebody's like, oh, you didn't say gonorrhea. I was like, no. So, yeah, there are lots of holes, but hopefully not enough that it all falls apart.
Okay, so with your permission, last question. And this is, again, also a great writing, a great article, 10 simple rules to becoming a principal investigator. And one of the things that you write there is, let me just quote, moving from doing someone else's research to getting other people to do yours. Being a PI is fundamentally different jobs to being a postdoc. And it's not like a graduate student or a doctorate, it's a postdoc. And postdoc usually has his own research that just happen to be in the same environment. And it's not an easy transition. And then you list like 10 simple rules about have ideas, publish papers, and many things as well. And now, many students and many postdocs that I know had very bad time switching from being a postdoc or even a successful postdoc to being a PI. And I want to just ask you about one thing. The first rule, which is have ideas. Now, how do you have ideas? Because I thought that being a postdoc means that you need to work in part on your ideas as well. So could you please elaborate just on one thing, because we don't have many time. Could you please elaborate on what you know about have ideas?
So I think what I was trying to say is, and you're right, so the postdocs have to have ideas, but often it's within the context of a single project, or they have ideas about how to move their single story on. You've kind of got to have a whole framework of ideas to support new people, to, some which will go nowhere. I think it's the engine on which our careers are, it's what we sell, isn't it? If we're a company, we're selling our ideas and the successful kind of development of those ideas. And I think without those, you can't run a group, because that's your main job in the group, is to come up with at least the initial idea. And it doesn't have to be right, but it has to be enough that somebody else can then go and build it further. So
how do you get the idea? Where ideas come from? Where good ideas come from? Yes.
So for me, it's conferences, and conferences with time to step back and think in between them, or some space after the conferences. So I get most of my ideas from talks rather than from papers, but other people get them from papers. I think you need to dedicate time to thinking about things, and to and to accept that you can spend a whole morning and have nothing to show for it. But that's not a wasted morning. It's just that day you didn't have anything to show for it. So it doesn't just happen. I think you have to put the time in, and then you also have to leave the gaps in between. I think your subconscious does a lot of the heavy lifting, but it needs to have the kind of input in, and then it grinds through it somehow, and then stuff pops out at the other end. So a range of different things. Exercise helps me as well.
I'm having tomorrow on the show Professor Barbara Oakley, and she is the author of like the most views massive open online course in the history, Learning How to Learn. And she heavily discussed, the difference between focused and diffused mode, and the importance of diffused mode in learning. Like wasted morning is not wasted, because something is built in your unconscious, and finally you get it in the shower, you get it in the conference, you get this aha moment, but this aha moment is built upon many wasted mornings. So
yeah. And it comes back to Stephen King with the writing thing's the same. Where he talks about writing with the door closed and then edit with the door open. It's that same. There is similarities in that creative versus editing kind of mindset in ideas as well, I think. So hopefully the two bits kind of feed into each other.
Professor John Tregoning, it was an extremely pleasure having you today. And you're not just a colleague in the context of academia and science, you're also a colleague in the context of book writing. And let me please hope that this will be your last book, and you will continue to publish science book for the audience, for the lay public. Thank you so much. It's been a pleasure. Thank you so much for your time.
Thank you. It's really enjoyable.
Okay, bye-bye.
Bye.